We have the human genome, that was published more than 20 years ago, and it's changed the way we do human biology. And we want to make that possible for all of biology. I'm Mark Blackster. I'm an evolutionary genomicist, and I'm a zoologist. And here at the Sanger Institute, the head of the Tree of Life program, which is sequencing all the eukaryotes, that's organisms with a nucleus, which live on these islands, which we call Britain and Ireland. At the core of everything we do is data generation. So we have the concept that large-scale data generation not only enables the answering of the individual little question we're interested in, but also many other questions later on. So it's going to be an open access library of life where you and anybody else can build their research on it. You can turn up and you can say, I would like to read the book of the badger. You could ask, why is it resistant to TB? Why does it carry TB? How is it related to other carnivores? You can use these data to screen ecosystems, to understand how individual species are responding to change, and to help breeding programs. I can see use in biotech, so generating new enzymes, generating new drug lead molecules, because many of the species we are sequencing have really interesting biology and really interesting biochemistry. One of the things we sequenced early on, it's a thing called an Emertian worm, and it catches its prey using venoms. And this venom works on shrimps and it works on little fish. So this venom could be very useful. Maybe you want to make a new anesthetic, maybe you want a new pain-killing drug. Another field that the data I hope will be useful in is understanding human disease. Many of the genes and proteins that we make, copies of them are also made by other organisms, and we can use those to understand what, if you like, are likely to be the damaging changes that you see in us in disease compared to what can happen. And by sampling all of life, we're sampling billions of years of evolutionary tinkering. So impacts in human health, impacts on bioindustry, impacts on evolution and ecology, and impacts on conservation science.
My name is Kerstin Hau. I'm the head of production genomics in the Tree of Life department at the Wellcome Sanger Institute. We are aiming at sequencing 70,000 species. So we have partnered up with many institutes that help us collecting those species. So they go through the country, they dive into the oceans and into the lakes, and they are getting samples of the species that we can then sequence. And we are extracting the DNA, so the building blocks of this species that make it what it is. And we are sequencing it. That means we are determining the individual letters on this very long string of letters that's in there. We can only do this in short bits, and that is quite a challenge. But the methods that are available and that we help developing are getting better and better. So we can now generate literally in a few hours what took us more than a decade to achieve in the past. We hope that the impact will be vast. Firstly, of course, for the research community, because they will now have at their fingertips what usually takes them lots of money and years to generate themselves. They can just go to our website, get the data, work with it. I believe that we are contributing majorly to, for instance, the development of new biomaterials, the discovery of potentially new medical agents, maybe even new antibiotics.